I'm Stefano Piotto, Professor of Chemistry and Bioinformatics and Director of the Biomaterials Center at the University of Salerno. I'm also the co-founder of a startup that uses artificial intelligence for drug design, SoftMining.it. Pharmaceutical industry is extremely inefficient, and this is not a provocation, but a fact. When an industry is efficient, the cost of each individual product decreases over time. In contrast, in the pharmaceutical industry, the cost of a molecule constantly increases over the years until it has exceeded $2.5 billion to develop an anti-cancer drugs. The number of approved molecules coming to the market decreases over the years, a decrease suited in recent years by the mere fact that the several orphan drugs have been approved. What are the reasons for this? There are two reasons. The first is related to the incredible complexity of the system we are looking at. We have recognized a typical biochemical pathway where there are many components, and each of them influences many other elements. The system is strongly feedback regulated. The second reason is that often incorrect models of activity of the drug are used. There is a serious epistemological problem with research and analysis. For example, more than 30% of genes do not have a phenotypic effect. This means that you do not notice any apparent change in the system if you knock down one of these genes. Why? They are part of a much more complex, redundant regulatory system. Even if you turn off some genes, the organisms can still compensate by following other biochemical pathways. If this is the case, it is clear that research and analysis procedures must be rethought. The drug discovery cycle is not efficient because it involves the identification of a specific target and then the design of something that interacts specifically with that target. What is typically done in the pharmaceutical industry can be summarized with the Latin expression ceteris paribus, which means to keep everything constant except the variable we consider. This only works if the variables are not feedback regulated to each other. If there are many variables but they are correlated, they cannot simply be held constant. Still, the answer is often to ignore this fact. Concentrating on a receptor, trying to modulate its activity is not the best way to develop a new drug. The active site of a receptor can be similar to many others. When the molecules interact with the receptors, we have what is mistakenly called off-targets. The ancient Romans also spoke about mutatis mutandis, which means considering all factors that interact with each other and the variable of interest. The models, therefore, need to be rethought. Here is the cover of a wonderful book, Gödel, Escher and Bach, by Douglas Hofstadter. The central object casts its shadow on three orthogonal walls. The operation of illuminating an object and interpreting its shadow can be seen as a metaphor for a scientific experiment. When we do an experiment, whether it is real or in silico, all we are doing is interpreting a shadow. For example, we subject the sample to stress, which can be a light beam, and analyze the output, in this case, the shadow. By reading it, we try to get information about what is the object of our experiment. Three different experiments give three different representations of the same object. And it is our work to try to build a model of that object that is compatible with the results obtained. When we say that the experiment is true, we refer to the projection of the object that represent reality. So a shadow is real, the other shadow is real, and this is the reality. What we have done over many years in different European universities is to update some models that are provably wrong. artificial intelligence is frequently used, and most people consider it as a sort of black box where large amounts of data are inserted. Traditionally, if you want to design a drug, you must know the structure of the target that can be multiple, not well defined, and whose concentration can vary over time. Oscillating systems are typical of any feedback regulated mechanism. This could mean that if there are many receptors in a cell, they will not have a constant concentration over time. It is important to know that target structure using experimental methods such as X-ray, NMR, etc. And beyond the fact that even these systems bring with them errors, we often do not have the experimental data because we have not yet acquired it, or because it is difficult to obtain in many cases. The wrong model is the idea that during folding, for example, the system's energy decreases monotonically, and therefore the native structure of the protein corresponds to the minimum of energy. This was an illusion derived from the Anfinsen experiment. Most proteins require assistance from chaperones and chaperonins. For example, the very famous Groyel-Groyes system. The native structure does not always correspond to the minimum energy. Today's most successful approaches for folding are AlphaFold or TR Rosetta, or the method developed by us that exploit the distance and the interresidual orientation. It's called Folden. We exploit that tool to analyze protein-protein interaction and to generate hierarchical folding, simulating the work of ribosomes. Considering the interaction of peptides with lipid membranes, we started to unveil the reason for lipid extreme complexity. We know about 10 nitrogen base. We know about few tens amino acids in nature, But if we consider the class of lipids, we are talking about several thousands. Why? What is the meaning of this large amount of lipids? The old model considered the membrane as a sort of wet bench of proteins. So just a space to support transmembrane proteins. But analyzing the different lipid composition of membrane, it can be seen that they also differ depending on the biological site. For example, docosahexaenoic acid is very abundant in neurons, or ceramide in the membranes of skin cells. The physical state of membranes changes continuously and adapts to the protein inserted into the membrane. So if we increase the temperature causing a heat shock, the effect will be visible both in the membranes and in the proteins. Increasing the temperature will lead to the unfolding of the protein and the increase of membrane fluidity. Even if we change the lipid composition, we will have an unfolding of the protein. Nevertheless, if we readapt the lipid composition after the heat shock, we can recover the full protein functionality. The various transmembrane protein sequences could then be used as a sort of bioinformatic probes to distinguish between one membrane and another. We have extracted and analyzed the transmembrane portion of thousands of proteomes, and we define a suitable metric to understand the interplay between transmembrane portions and membranes. Through the analysis of these different portions, we were able to reconstruct a phylogenetic tree based only on the differences between the transmembrane portions, and we were able to divide the archaea from bacteria and the eukarya. On the right, most importantly, we can see how different organisms living in extreme conditions, such as thermophiles, are clustered together, even if they have different classes and are phylogenetically very far. They will therefore have different proteomes but similar transmembrane sequences. This was the very first demonstration of coevolution in cell membranes. Thus, we were able to identify a series of bacteria that became a sort of mine of possible sequences that can be tested as a biocides, the so-called antimicrobial peptides. As a side effect, this approach, that permitted us to develop a new metrics to analyze peptide sequences, allowed us to develop a new metric to compare strings in general, so a universal method to analyze strings. We developed two search engines, Arise and Brevia, on the SoftMining calculation computational platform that measure the semantic distance between two documents. This is the same approach we use for bacteria. Arise analyzes scientific articles, and the search is fully semantic and outperforms Google Scholar or Scopus search engines. Brevia does the same job of Arise, but on patent literature. The semantic analysis combined with the artificial intelligence has also been used to analyze trajectories of molecular dynamics. Molecular dynamic trajectories are mapped into strings, and this permitted us to discriminate, for example, between agonist and antagonist, a distinction that could not be done only on the basis of binding efficiencies. Another application of our metric is in the calculation of the similarity between molecules. If we ask to cluster these 4 images, they would probably be separated into 2 amphibians, a bird, and a woman. Actually, if you look carefully, you can recognize that the 3 images are just human bodies painted. It might be very complex to try to determine the similarity between 2 different objects. In this sense, an approach that we used was the persistent homology to determine the similarity between active sites, obtained by inserting water molecules that are reorganized into a network of hydrogen bonds. And given this network and this graph, we could also reverse engineer the possible best molecules to interact with the binding site. The software is called Matisse in honor of the painter and is available on the computational platform of SoftMining. After that, reverse engineering work was done to design new ligands and determine how far apart different active sites are. This is most useful for repurposing and for understanding potential side effects of a drug. The molecular similarities are also calculated with a new dedicated electrotopological parameter. It works taking a molecule, you can see in the upper left, and calculating for each atom a certain property. For example, you can take into account the mass, the charge, the polarizability, or any kind of quantum mechanical calculation, even frontier orbitals. And then we can map this property, casting the shadow of the molecules onto the sphere that envelops this molecule. And once we have this mapping, this 2D mapping on the surface of the sphere, then we can start to calculation the difference in the different mapping, so in the different shadows of different molecules. This consists in a 3D to 2D mapping. The calculation of these matrices differences permits quantifying electrotopological distances between molecules more accurately than E-state descriptors. So actually, this is the best to date electrotopological parameters to describe the quality of a molecule. Compared to traditional parameters, this allows determine much more accurately the distance between 2 molecules. Finally, we developed a method to calculate the best interaction with a cluster of receptors rather than a single one. starting from a molecules to be optimized, we see how it interacts not with a specific target, but with an entire interactome. So it can reproduce the right pattern of activation and have a good efficacy-toxicity ratio. I've told you a lot in just a few minutes. We are available for any clarification and collaboration on integrating artificial intelligence into the drug discovery industry. Feel free to write me at Piotto, Piotto, @unisa.it. And thank you for your attention.